Today we're going to talk about mitosis. And what I've drawn out here is a generic cell. And we have already talked about this cell structure for the first test. As you recall, we have a nucleus. Here I've shown the DNA in the chromatin form and the non-condensed form as blue. And here we're going to say that DNA has been replicated. We also have our rough endoplasmic reticulum, our smoothie, our Golgi body, and a smattering of mitochondria. Now, those are really not going to become important for this discussion. I just draw them to give us some context. So what I'm going to do now is I'm going to remove those organelles. And I do so just so we can have a better palette to draw on. Don't have the idea that these organelles somehow disappear in this process. But for sake of completion and just for sake of discussion, I'm going to take those organelles out right now. Okay, so now the organelles have been removed. And again, don't have the idea that they somehow magically disappear. I've just done this to give us a better palette to talk about this, because we really want to super focus in to what's happening in the nucleus. Now, what are we trying to do here? We are trying to make one cell, okay, into two separate cells. So we're trying to take one cell and replicate it into two individual cells. Now, preceding this, we replicated our DNA, so we now have a copy of our DNA in the nucleus. So we have two problems here. One, our DNA is in a very loosey-goosey state, if you will. It doesn't really move around very well. It's very exposed in this state so we can get to it and do things with it, but it's not in a good state that we can move it around. Okay, it's not in what we call a condensed form. So the first step we're going to do is to condense our DNA. And condensing, again, is we are wrapping our DNA up very tightly. Again, the analogy we used in class is think about your power adapter to your laptop, okay, or your lights at Christmas. When you have all your lights out at Christmas, you have them all, out, extended out, right? You have your power adapter to your laptop. You have it unwound. You're using that whole length of the power cable, right? Well, when we are using our DNA and making protein from it, that DNA is somewhat unwound in the nucleus. However, when you want to move your laptop, what do you do with your power adapter? You probably take the power cable and wrap it around itself, right? Because that's good for transport. Okay, how about your Christmas lights? You take a piece of cardboard and wrap those Christmas lights really tightly around that cardboard, right? Because that's a good way to transport it, isn't it? Well, same thing is going to happen here. Our DNA is in a very loose state. It's nice and exposed so we can make protein from it. And in this form, it's not very good for movement. So one of our first steps is we're going to condense our DNA. We're going to get it very tightly wound up and form those chromosomes that you are familiar with. So now we have condensed our DNA, and we have 3 chromosomes. And each of these chromosomes has a sister chromatid. So this is our first chromosome here, our second chromosome here, and our third chromosome here. Now, do you see the difference here? Is the DNA changed on a base level? No, not really. All we have done is we have wrapped that DNA up very, very, very tightly. We have condensed the DNA. And this is what we call the DNA condensing. Okay? Now, why is this good? Because let's go back to this idea here. We're trying to make one cell into 2, aren't we? So don't you think we probably want to move our DNA such that part of the DNA goes to the left and part of it goes to the right? If it was in that very loose state, it would be very hard to transport around. But in this condensed state, it becomes a lot easier to transport. So now we've got our DNA in a nice condensed form, and that's fantastic. But we're missing something here. We can agree that having it in this condensed, very tightly packed form makes the DNA very easy to move around. But what's the problem? Well, in this phase, how do we get to the DNA? How can we grapple onto the DNA with our microtubules to pull it apart? And the answer to that is we can't. Okay, so in prophase, the nucleus will start to dissipate. Okay, so now that nuclear envelope has dissipated. And this is good because now we can grab on to the DNA. We can grab on to these chromosomes. Before they were encased in a nucleus, we couldn't easily get to them. What we have now done is what we call prophase. Okay, so what did we do during prophase? First, our DNA began to condense. It got very, very, very tightly wound. Second, that nuclear envelope started to be removed. It started to dissipate. And why is that? First, our DNA has to condense so we can easily maneuver it around. It's physically much stronger now. Second, why did that nuclear envelope dissipate? Because now we can access the DNA. We can put things on the DNA to pull it apart. Before, we couldn't do that. And again, go back to the whole paradigm of what we're doing here. We're trying to make one cell into two cells. Okay, so prophase has accomplished two tasks for us, but we're not there yet. So next is the metaphase or transition to metaphase. So the first part of the transition to metaphase is microtubules of the spindle organizing center are going to connect to our chromosomes. Okay, these are going to pull apart our chromosomes. So first we have connected them, and this is what we'd call a transition to metaphase, or you could include this in the metaphase step. And next, these are going to move our DNA until we form what we call a metaphasic plate. Let's do that now. Okay, so now we are entering into the later stages of metaphase. And what have we done? We have used these microtubules. The red are our microtubules here. We discussed these as a part of our cytoskeleton in the first section of the class, or the first test. And these microtubules have maneuvered our three chromosomes, chromosome 1, 2, and 3, to somewhat of the middle of the cell here. Now, going back to our prophase, what happened to our DNA in prophase? So let's go back to this stage right here, prophase. What happened to our DNA? It condensed, right? Do you see why we need to condense our DNA? Now we can maneuver that DNA around the cell a lot easier because it's very tightly packed. What else happened in prophase? The nuclear envelope disappeared. Do you see how we can now access our DNA with these microtubules, what's shown in red? That's the point. Okay, so now we are in metaphase, and we have maneuvered our chromosomes in such a way that they are somewhat spanning the middle of the cell in almost like a plate or a linear line type of formation here. The next step is anaphase. And before we begin anaphase, let's go back and say, what are we trying to do here? We're trying to make one cell into two cells, right? So what's going to happen is our microtubules will depolymerize. They're going to start breaking apart at the ends. And when they depolymerize, these microtubules will become shorter. These will become shorter in this direction. And these microtubules will become shorter in this direction. Okay. You ever break open a wishbone? You know, you get a wishbone and two people hold on to it and both pull in opposite directions and it breaks apart. Well, we're basically going to do the same thing here. Okay? And in the anaphase step, right here, number 3, in the anaphase step, these microtubules will come shorter. Okay, they're depolymerizing, they're breaking apart. And as they depolymerize and break apart, they become shorter. These microtubules over here on the right become shorter in this direction. The microtubules on the left become shorter in this direction. Any guesses as what's going to happen? Well, if you guess it's going to break DNA apart, you're correct. So what's happened now in the latter part of the anaphase step is that these microtubules, because they are depolymerizing and becoming shorter in these directions, they rip apart those sister chromatids. And now we have separated our DNA. Now, do you see why we condensed it in prophase? Do you see why we removed the nuclear envelope in prophase? Do you see why we lined it up in metaphase? It was to get to this anaphase step to separate our DNA. And again, these red lines I'm using here represent the microtubules. Those are depolymerizing. Those are becoming shorter at one end, and that shortening pulls apart our DNA. Now we're on our last part, which is telophase. What has happened here? Well, our DNA has moved to the opposite ends of the cell, right? Think about your first section of material. We said that the nucleus protects our DNA, right? So very rapidly, the cell will want to reform a nucleus surrounding that DNA because that DNA is like gold. And we don't want to just let it just sit out there without a nucleus in our cells. So very quickly, our cells will begin to reform this nucleus. Also, the DNA, it has begun to decondense. It condensed in prophase for what reason? So it was nice and tight together so we could easily maneuver it around and separate it. Well, that job is done. We're done with that. So now our DNA begins to become more loosey-goosey, if you will, becomes to decondense so we can actually use the information there. Okay? Now, the steps after this will be the cytokinesis, where this cell rapidly begins to divide into two separate cells. Now, different mechanisms are done with animal cells and plant cells. Those will be covered in class. But long story short, this cell will very rapidly become two cells. Okay, so now our one cell has become two cells. And that was really the whole point of this operation, wasn't it? We're trying to make one cell into two cells. So let's go through the major points here of what we've done. First was our prophase step. What major two things happened in prophase? Well, first, our DNA began to condense because we're going to move around the DNA. So we need to have it in a very tightly packed form. Second of all, our nuclear envelope started to dissipate because we needed to access the DNA in the later steps, right? We need to move it around with microtubules. If it's in a shell, that nuclear envelope, how can we get to it? And the answer is we can't. So we had to get rid of that nuclear envelope. Second step was metaphase. What happened with metaphase? Well, first we used the microtubules to attach to those replicated chromosomes, right? Because we want to pull those apart, but first we have to attach them. And second of all, the microtubules depolymerizing and shortening, they maneuvered the DNA such that it was in a linear row. Metaphasic plate. What happened after that? Well, those microtubules began to really depolymerize and shorten. And they depolymerized opposite to each other such that it pulled the DNA apart. It physically wrenched it apart. Think of two Jeeps on each end of the cell and their winches, which are those metal metal cables that pull really tight, pulling apart the DNA. That's what's happening. So in anaphase, we ripped apart the DNA such that half of it went to this side and the other half went to this side. Okay? Telophase, what happens? Telophase, the whole thing is done, right? We start to reform that nuclear envelope because, as we said, that nuclear envelope protects our DNA, and our DNA is like gold. Okay? Second of all, what happens is our DNA begins to decondense. It's done being all tightly packed. It starts to go to that less condensed state so it can actually be accessed and made into proteins. Okay, and we talked about mRNA and protein. And those are the 4 steps of mitosis.